Hello, in this video we will discuss about the brain derived neurotrophic factor signaling pathway. So in this way, what is this? This is the different topic we will discuss in this video. That is the brain derived neurotrophic factor. Basically, it is the brain derived neurotrophic factor is stimulated via a necrotic factor kappa B, and this necrotic factor kappa B which that is involved, and to necrotic factor kappa B will stimulate the nitric oxide production. So it means the vasodilation as well as other things due to the nitric oxide production, but here we will not just understand that. So here is the BDNF is the neurite outgrowth. So it means the neurite outgrowth, a nervous system growth, and through a activation of JAK, STAT, RAC, and CDC4-2 pathway. So on the other hand, the brain-derived neurotrophic factor enhance the oxidation of fat through AMPK-mediated inhibition of ACC, and it also play a role in microtubule assembly through inhibition of glycogen synthase kinase 3 beta enzyme. So remember that is a BDNF is used for the development of the brain and development and different neural pathway which that is increase the memory, increase the learning process, and increase the cognition as well as the increase the alertness related to the learning. So here's the pro brain derived brain pro BDNF, which that is the presynaptic neuron which contain vesicle, and vesicle also contain proteases. These proteases will supply this BDNF to become a pre-peptide as well as the pro BDNF. So the pre-peptide form, and after the splitting, so the both type will be occur, and this will be exocytosed through vesicle. And here is the receptor. This receptor is known as the TRKB. We will discuss later in this video, and it is high affinity, and it can be uptake the BDNF. So this BDNF will be uptake, and after this BDNF will be uptake, the autophosphorylation will be occur. So here is the P75 anti receptor which that is contain low affinity and bind with the pro BDNF, while here is the BDNF will bind with a high affinity with the TR KB is the receptor we will discuss later, and autophosphorylation will lead to cascade of phosphorylation events will lead to produce the MAC1 and 2 and PI3K pathway. So this is the postsynaptic neuron. So let's understand with the detail with the signaling pathway in the cell. So here the synapse occur, and this synapse where the synapse, so this is the talking to each other the neuron. So the BDNF is a neurotrophin essential for growth, differentiation, plasticity, and survival of neurons. What is the plasticity? Plasticity mean? Plasticity means the nerves will be more protect as well as they create the different pathway for the nerve impulse and integration. So it is also required for process such as energy, metabolism, behavior change, and mental health, learning and memory, as well as stress and pain and apoptosis. These all things also required for the BDNF. So remember the improvement of the memory due to the BDNF production. So the BDNF will bind with the nerves, will stimulate and more growth of the nerves, and either the nerve damage will be apoptosis. But remember here is the cell which that is I am drawing the bigger picture, extracellular matrix and plasma membrane and intracellular matrix in the form of cytoplasm, while here is the nucleus which that's contain nuclear membrane, contain nuclear pore, but here is I am not drawing. So the nucleus, and here is the DNA, which that is the DNA is the genome, the genetic material of the cell. So remember the DNA is used for the transcription to produce the messenger RNA, which that is used for the translation to synthesis of protein. So here the extracellular domain contain tyrosine kinase receptor. This tyrosine kinase receptor is a dimeric, remember. And important thing you should need to understand that during fasting it will be enhanced. So remember, during the fasting the BDNF will be increased. And extracellular domain contain tyrosine kinase receptor, the dimeric, and it is also known as the tropomyosin related kinase B. So that is why it is known as the TRKB. And basically it involves the central nervous system and peripheral nervous system expression. And the fasting will trigger the neuron repair and the cognition by different synapse pathway. So improve memory, improve behavior, and improve learning through these BDNF signaling pathway, and it will improve the nerves. And remember, this is the meaning of these, the plasticity, mean the protection of the neuron. So remember, here is the binding of the BDNF. After the binding with the extracellular domain, autophosphorylation will be occur inside in the intracellular domain, and the autophosphorylation with the phosphorylated site. And in this way there are different the docking of the different protein with the phosphorylation. For example, son of 7 less will be bind after with the SHC binding with the phosphorylation site, and after this dimerization will be occur, and in this way the phospholipase C gamma will be bind also. And the other side is the son of 7 less protein will bind with the SHC and the GAB1. So GAB1 will be bind, and the growth retinoblastoma protein 2 will be bind with the source. And in this way, remember that the GAB1 will bind with the phosphoinositide 3 kinase. After this phosphoinositide 3 kinase phosphorylation will lead to other phosphorylation events. So here is the IP2 are present in the plasma membrane will convert with the phospholipase C gamma to produce diacylglycerol DAG and IP3. So the IP3 is used for the calcium efflux from the endoplasmic reticulum. So remember that. And here is the protein dependent kinase 1 will stimulate the AKT via a stimulation by a PI3K pathway. So this is the AKT will be stimulated. So here is the phospholipase C gamma. Again I am drawing with the IP2 converted to the IP3, and diacylglycerol will be separate, and the protein kinase C activation and the enhancement, and in this constitution of the calcium will be increased through efflux from the endoplasmic reticulum when the bind with the IP3. So the cyclic AMP will be CAMPK pathway will be increased. So remember, here is the neuroplasticity and survival and growth will be occur due to these pathway. And here is the AKT will be inhibited by the 10 protein. But in this way, remember that during the fasting state, the mTOR pathway will be inhibited. So in this way, it will be stimulated, while the mTOR pathway will inhibit this function. And here the survival, the neuroplasticity and survival of the cell and growth will be occur. And the brain derived neurotrophic factor will also produce more, and the cytoplasmic neuroplasticity will be increased. Anyhow, here is the son of 7 less will bind with the RAS, RAF, and MEC pathway, so the mitogen activated kinase pathway. And in this way, the ARC pathway, and in this way, the both MEC and ARC can be inhibited by a MKP1 protein. So this is inhibition mechanism. But in this case, here is just remember that how to produce. So it will become a transcription factor ARC and bind with the transcription site the specific gene to produce the messenger RNA. This messenger RNA will produce that protein which that is reliable and comfortable for neuroplasticity and survival of the growth and survivalship. So thanks for watching. Please make sure to subscribe. Bye.